speaker, Dr. Garry Rosewarne, SRA General Manager of Variety Development. So Garry leads the variety development team and is a highly experienced blend breeder with an impressive career across both Australia and international research organizations. Throughout his career, Garry has led numerous successful R&D initiatives, including the National Lentil Breeding and Lentil Breeding Infrastructure programs, jointly funded by the GRDC and Agriculture Victoria. He has held senior research positions with CSIRO in Canberra, CIMMYT in Mexico and China, and currently serves as the president of the Pulse Grain Society of Australia. Garry's previous research focused on leveraging data to drive strategic breeding approaches and introduce novel quality traits into pulse crops. But thankfully, Garry eventually came to his senses and made the wise decision to move to the greatest crop in the world, our beloved sugarcane. So without further delay, and because I know you are all eager to hear about the SRA 5 Years Variety Development Program, Garry, the virtual floor is yours.
Thanks so much, Natalie. I'll just try and share the screen, and we just better update that little blurb at the beginning, because I did relinquish the presidency of Pulse Grain Society of Australia. I'm not longer doing that anymore. Moving on to sugarcane wholeheartedly. So welcome everybody, and thanks for coming to this seminar this afternoon. I'm going to give an update of where I think that where I want to take the variety development program. The board has charged me with looking at developing a 5-year improvement plan for varieties, and it kind of goes hand in hand with the team that's developing our strategic plan as well at the same time, a more broader SRA strategic plan. Now there, where am I? Here we go. So the outline of the talk today, I'll just sort of have a bit of a look back from where we've come from in variety development, look at some historical production figures, and just talk a little bit about the difference between defensive and offensive breeding and what we've done in that in the past. And then move on to how we can do things better, and that'll cover areas such as incorporating genomic selection in the program, high throughput phenotyping, what our current thoughts around genetically modified crops are, cutting edge statistics, understanding genotype by environment by management practices and how that impacts on our breeding program, and then work that I'm interested in trying to better align the breeding program with industry, and maybe potentially a new concept we're coming across around fast track breeding or reinvestigating fast track breeding. So the graph you see on the left here is the total production figures in the blue columns in Australia going back to the mid-1980s. And there's a blue dotted line there as well. And that's kind of, that shows a moderate increase over time, going about 1 or 2% per year, I think. So we're really not increasing production in the landscape very much. The orange solid line is tonnes of cane per hectare, so it's on this y-axis over here. And you can see it jumps around a fair bit, but the regression line there is quite flat. So what that is suggesting is that we haven't had any increases in tonnes of cane per hectare for a long time, or over this 40-year window. And so that's probably a bit of a concern, and people wonder why that would be. What the breeding program has done is had to deal with several major incursions of diseases. So Pachymetra was first identified in the early 1980s, and the breeders went away and bred for resistance for that. Orange rust resistance in sugarcane in Australia broke down at the turn of the century, and you can see there was a dip in tonnes of cane per hectare and also a dip in total production. Now, whether this looks like a correlation with the timing of the orange rust outbreak. A correlation is not always causation, but it's interesting that happened. And then about 10 years later, we had smut come in, and again there was that dip in productivity. And so these three major incursions, and that really puts challenges to the breeding program, because every time you get a disease incursion you've got to breed for, you've got to bring in a lot of exotic germplasm, and that lowers your yield potential in the breeding population. And what I've said here, it really puts a handbrake on really pushing forward yield gain. So you can see my sports car racing through the sugarcane fields with its handbrake on. Not the best thing. But also one thing you'd also notice about this graph is that you look at that orange line that's jumping up and down in tonnes of cane per hectare, and it's getting narrower and narrower. And maybe that's a reflection that we're getting more resistant germplasm into the growers' hands, and that actually gives us yield stability. And yield stability is an important component for a grower to consider. So when we breed for disease resistance, we've done a lot of good work in SRA over many years and done yield loss studies. And so this first column here is like the disease rating of different varieties. And if we've got a variety that scores an 8 or a 9, and then we have an orange rust epidemic, that variety could lose between 20 and 40% of its yield. So disease can really have a major impact on yield. Smut's even worse. And so then when we try to look at breeding thinks, oh, it's all about getting higher tonnes of cane per hectare, getting better sugar and stuff. Now, if we run a really good breeding program, maybe we can go along at 1.5% a year. Over 10 years, we think, wow, good breeding program has increased the productivity by 15%. In today's language, in today's industry, that would be worth about $250 million. It's pretty good. But when we look at these defensive breeding strategies, if we were to have release all varieties that were, an 8 or a 9, we would be losing virtually half of our productivity. So you can see the defensive breeding program processes are really important in maintaining a viable industry. And you can see what's happened in the breeding pipeline. This is the percentage of clones in our FAT trials, our stage 3 trials that have resistance, and over time you can see the levels of resistance in smut have steadily increased. And this was brought out at a recent industry meeting. I went down to Bundaberg for their annual awards meeting, and they claimed that 99.97% of varieties that are grown in the southern region have smut resistance. So that's a great outcome. And if you don't believe me about these figures, about how destructive that might be for the industry, I've just put on a graph here about international production figures. So here Australia is at 85 tonnes per hectare, and you look at a lot of our neighbouring countries that don't really have good breeding programs or don't breed for resistance, and they're between 40 and 60 tonnes per hectare. And I think you'd be aware that if Australia was only producing 40 to 60 tonnes per hectare, we would not have an industry anymore. And just to highlight a bit further, this is some data from the central region. So this is looking at the varietal composition over the different years, going right back to 1994. And you can see back here in the late 1990s, we had this variety Q240, and at one stage, 87% of the whole region was grown to Q240. Then orange rust broke down, and pretty quickly, Q240 was no longer in the system anymore. But equally as quick, it was replaced by other varieties that were already in the breeding pipeline. And you can look at their tonnes of sugar per hectare here. There's Q240, and the growers didn't really take too much of a hit when they swapped over to the new varieties. That's great. And here's the key around the orange rust susceptibility and the resistant ones coming through. And that also carries forward into the future as well. One of these varieties, Q157, was released in 2001, orange rust resistant, and it started to get a bit of a foothold, but then smut came into the industry in the East Coast. And so very quickly we had to replace 157 and these other varieties, and Q208 came out first. It was intermediate to resistant for smut, a bit of a step up in tonnes of sugar per hectare, and that was followed by several other varieties, Q183, Q240 and Q253, and you can see they've now combined orange rust resistance with smut resistance and a higher yield potential. So that's really what we're talking about when we say defensive breeding is really important for the industry. And then, so I've talked about smut and a bit about pachymetra, a little bit about orange rust, but there are all these other diseases we're trying to breed for. And so sugarcane is particularly challenging because I've worked on wheat and in pulse crops of field peas and lentils, and I've never seen a crop that has quite so many diseases. And I guess that's what happens when you grow crops in the tropics. There's plenty of things that want to eat them. Now let's change tracks and talk a little bit about how breeding works and how we can measure our success. So here is the classic bell-shaped curve or a normal distribution. And so what this says is, on the x-axis we've got tons of cane, and on the y-axis we've got the number of clones that fall into each of these sort of yield potential levels. And so I'm saying maybe this is an illustration of our CAT trials. So we've got a couple of thousand entries in our CAT trials, and the vast majority of the CAT trials fall around this middle area of the normal distribution. And then there's a few up here at the high end and a few down there at the low end. And what we want to do in a breeding pipeline is we want to select the ones at the high end. We grow them again next year. And then we can say, well, the difference between these two peaks is how we've made some sort of level of genetic gain or yield improvement. And then from then onwards, we'd want to try to select these really elite ones out here, a couple of years of yield data now, and maybe they could come on to be some of the new varieties coming through the pipeline. So that's a concept around breeding. We can measure it in another way. So again, this normal distribution, and this is from, say, the fat trials in 2021, and then next year we grow our fat trials and the germplasm's advanced a little bit through the breeding pipeline, and hopefully that normal distribution moves to the right in 2022 and further to the right in 2023. So we're always trying to get the population moving forward and getting higher yield potential or better disease resistance or better quality traits. And we measure this in the breeding pipeline in our fat trials. And so here's a graph of that peak there for each year going right back to 2007 and coming forward. And you can see that the slope is going up. Doesn't look like it's a really high slope, but it's about 0.5% per year, which is quite reasonable for a breeding pipeline. There's also another way we can measure genetic gain, and that's by looking at the year of variety releases. So I've just taken a bit of a snapshot here of a number of varieties released in the north over time. There's their name, and that's tonnes of cane per hectare compared to Q208. And I've graphed that over here. The graphs, the dots are a bit looser around the regression line here, but you can see that we've always, the more recent varieties tend to trend to have higher yields than the ones that are older. So that's what we expect in a breeding pipeline. And that one there is increasing at about 1.5% per year, which is pretty good. And I saw a podcast late last year. They'd done some work in wheat in Western Australia, and the breeding over there thinks they increased their productivity by 0.6% per year. That's wheat with about 10 times the resources that SRA have. So if we're doing 1.5% per year, we're doing pretty good. And then we can put I've done this for all of the different regions, don't worry about all the graphs, but we've looked at REGV, tonnes of sugar per hectare, tonnes of cane per hectare. That's the north graph. But then I guess this is the number we want down here, the average overall, and look at tonnes of sugar and tonnes of cane. On average, we're increasing genetic gain by 0.8% per year. So I talked about wheat doing 0.6, we're doing 0.8. This is solid, robust data. This is meticulously grown yield trials, good statistics run over the top of it, hand on the heart, this is what we've done within the breeding pipeline. We also do a whole bunch of quality testing too. So I've talked about disease testing, now I've talked about yield. Quality is also important. So we measure over 16,000 samples per year for commercial cane sugar and fibre quantity. We mill these, we extract the juice, measure those parameters through NIR, and every 10 sample we test with, we validate with wet chemistry. We measure other components of the fibre shear strength that measures fibre toughness and resistance to processing, impact levels as well, how brittle the cane is, and we end up drawing these graphs down here for both shear strength and impact. You don't want things that are too high and you don't want things that are too low, so we try to get varieties that fall in the box, within the box here, and all our new varieties have to have this sort of quality testing before they'll be released. Also on this graph there's a fibre length. If the fibres again are too short, short fibres improve milling efficiency, but too short they gum up the mill, and too long it results in higher milling costs. So again, we want the dots that are blue here as well. So very detailed testing. We also do some crystal quality testing that enables Australia to access some premium markets. Quite a unique thing with Australia, but the benefit to cost ratio on that investment's been 159 to 1, which is quite astounding. And so we've talked about the disease, talked about yield potential and quality, and then where do we stand in the world? Well, here's the percentage production of all the leading countries that produce sugar, and Brazil's right out in front. 42% of global market comes from Brazil. India's coming second, and then you've got the next three, Thailand, China and Pakistan, and down at about 8 or I think 10 at the moment, Australia's down there at about 2% of global production. So we're quite small players on that. But how do we do? when we look at this. So this graph here, the solid lines are just the total hectares that each country plants every year. Australia's not on this graph because we're right down the bottom here at about 300,000, and that's pretty consistent over that time period. But the dotted lines show our tonnes per hectare, and as I said before, our tonnes per hectare is pretty constant at about 85 tonnes per hectare going back 40 odd years. But you'll notice there's a gap in front of everybody else. And it's only just recently that Brazil and India are starting to get up to our levels of production. And it'd be interesting to understand the mechanics behind that. This is production in field. Some countries like Pakistan, they've got quite a steep growth, but we know they're going into a lot more irrigated areas too. So it'd be great to understand how these levels are rising. I know in Brazil it's probably because the mills actually have a lot of control over their farmland, so they can really optimize their agronomic production in that regard. So we're always up there, but we've got to stay ahead of the game. So how do we stay ahead of the game from a variety development point of view? The industry's got major headwinds. Sugar prices are down quite a bit. The industry is struggling. We don't have any subsidies in our agricultural industry, and a lot of other countries do. A lot of other countries going into irrigated areas and so forth. And our industry seems to be fairly stable, but with these headwinds. So there are some technological advances we can bring into the breeding pipeline through genomic selection, high throughput phenotyping, genetic modification, and statistical method improvements. But then also, how do we do our field-based research? And so part of the stats will give us a better understanding of genotype by environment, by management interactions. This is trying to find a genotype that's fit for a specific environment and really characterising those environments a whole lot better. I want to focus on better alignment of the breeding strategies to farmer practice. How can we work better within SRA to get better agronomic packages out to growers? And I also want to provide you with finish on a couple of concepts that have just come across my desk fairly recently, the farmer participated plant breeding and whether we can really reduce the breeding pipeline down significantly in a number of years. And they're a little bit questionable. We'll go through them and see what the options are. So what's this genomic selection? It's a method that can efficiently predict values of traits in new clones. So you can imagine if we've got a whole bunch of seedlings, we grow 100,000 new seedlings a year. Of a percentage of those, we could take a leaf sample, extract the DNA from that, and from that DNA get a prediction of yield, CCS, fibre, smut, and pachymetria. And we can do this now. SRA has invested in UQ over the last 7 years to develop this technology, and now it's mature enough that we can start to deploy it. So you genotype once and predict for many different traits. That's what we can do now, but what we really need to do, it costs about $100 to get the genotype from one clone at the moment. But what I would really like to do, if we're genotyping once, what other traits can we get? And I'm really keen to get an understanding of the genetic architecture behind ratooning, or some of these other diseases like red rot, Fiji, leaf spot, RSD, mosaic, and nematodes. So we can develop these, what they call these prediction equations for all of these traits. And then further down the track, I'm also keen to look at some of the physiological traits that interact with agronomic practices, things like tillering, plant height, maturity, lodging, stem thickness, trashing. If we can get all of this whole package together with predictions, we can genotype a seedling that's never seen the field before and get some level of understanding of how it will perform in all of these situations. So this technology is an absolute game changer for a breeding program. So this is a schematic of the breeding program at the moment. We start with crossing. We grow our stage 100,000 stage 1 clones and propagate them. We grow our stage 2 trials out to stage 3 propagation, stage 3 trials. And then finally we get down to this product development phase where we go accelerate max prop and hopefully release a clone. So it's all a process where we start out with 100,000 clones and maybe 10 or 12 years later we might release a couple of clones. The breeding cycle, as we call it, is how long it takes from when you make a cross to when the progeny of that cross is available as a new parent. And so that's about 10 to 12 years in this sort of cycle. That's a standard breeding program. and you'll see this style of structure in many breeding programs of a whole range of different crops. So how do we apply genomic selection? Well, the first thing that genomic selection allows us to do is to select parents much earlier in the breeding pipeline. So I've said 1 year, probably going to be 1 to 2 years in our hands. So rather than waiting for 10 to 12 years to select a parent, we only need to wait 1 or 2 years. So what does that sort of mean? And when you look at my age and you're looking at your superannuation and every year you're hoping to get another 10% increase in your superannuation, you're going to build that up and you're going to retire at the same level. Well, what this sort of system talks about doing is, could you imagine if you got that 10% every 2 months instead of every 1 year? And imagine what your superannuation would look like when you get to 67 and you're going to retire anyway. So that's kind of how genomic selection works in the breeding construct. I just had a message that people can't hear me. I'm not sure what I can do about that, but I'll just keep on powering on unless someone else complains. There's another, maybe I'll speak a bit louder. There's another,
sound is good.
Okay, good. There's another area where we use genomic selection, and that can be in actually selecting the clones we go forward, right? So we've got 10,000 clones here that I plan to genotype, and really when we make this selection down to 7,500 and even down to 2,500 clones, we don't have much information. The only information we've got is really one yield trial at one site, and that's not really robust data for us to use, but we do it anyway. But if we genotype here, suddenly we can get access to predictions for all of these different traits. And that's quite phenomenal. And so when we talk about breeding and how we can move the dial in a breeding program, we talk about two concepts around selection intensity. That's going from 10,000 clones to the next step of 2,500 clones, but also selection accuracy. And that's how accurate is our phenotyping at this stage. And you can imagine we don't even do a lot of this testing, the SMART and the pachymetra returning we don't do until down here. SMART and pachymetra we don't do until around this sort of stage here. And so really we don't have any accuracy for these traits at all. And so suddenly we're going to have access to all this sort of stuff. So it's really a profound way we can move forward. So I'm really keen to get it embedded in the breeding pipeline. We can also do something, I talked about this early stage up here, how important that is in really supercharging the breeding program, but we can actually do more than that, just that, because we can start to use AI tools and what we call computational breeding. So if we've got genotypes of these 10,000 new clones, we can simulate what would happen if we crossed any two of those individuals together. And we can simulate what the progeny would be, and we can simulate for all of those different traits as well. And so it's really getting into really big data now, and then simulating potentially millions of crosses. We only do 500 a year, so we can really pick the very best crosses to really give us the best outcomes. We've started to invest in this already. So this is our new DNA extraction robot. And this is the scale of what we plan to ramp up. We've been genotyping a number of clones, but within 4 or 5 years, we plan to do 10,000 a year, and that's probably where the budget maxes out at this stage. Then changing tracks from genotyping and now going to phenotyping. So phenotyping is just what we see in the field. How tall is a plant? How many tillers does it have? What's its response to a disease? That sort of stuff. And you can see we've got a team of staff out there, and they're collecting samples, and they're going to go off to the juice lab, and so it's all very labor intensive. So what can we do? There's several different phenotyping technologies. The workhorse, and one we're deploying actively, and again have had active research within SRA probably for the last 5 or 7 years or so, around capturing images through remote sense drone technology. There's also this sort of technology, the LAMP assay. It allows you to do things like disease assays in the field, and this micro NIR that we're trying to develop to see if we can measure characteristics around sugar content in the field. But I'll just focus on the drone work at this stage. There's multiple sensors we can put on, and the different sensors do different things. So visible, that's just like putting a camera on the drone, and we can get measures of height from that, plant count, cover and gaps, flowering, lodging, that sort of stuff. You can also do thermal imaging, and that allows you to identify areas of stress in the field. Multispectral gives you vegetation indices related to chlorophyll and so forth, and then hyperspectral sort of gets inside the leaf and actually gets to subcellular sort of levels, and we can get information from that as well. So all these different sort of technologies are in our hands currently. Then, so we're starting to generate a whole lot of data. How do we interpret it? And really what we want to do is combine this sort of technology with the genomics processes that we've got in-house. So really what we want, what we're the current state of play as, we look at all of our yield data from 2011 to 2024 in all of our fat trials. We've got about 8,000 clones in all of those, and we've genotyped them. So that 8,000 clones will give us about 500 million genomic data points. The yield trials and all the traits we collect from them, that's another 126,000 data points. Then you put in the SMUT and Pachymetra scores, that gives us our 5 prediction equations that we've already got. Where to now? By 2031, we want to be genotyping another 10,000 clones a year. I mean, it's taken us 10, 15, 13 years to genotype 8,000. We want to be doing 10,000 a year. So another 500 million data points every year for that. We're currently doing about 15 flights a year. We'll continue that and make sure all that data comes into our breeding pipeline. We want to do multiple diseases. I said we've got SMUT and Pachymetra, but I also highlighted all those other diseases we've got. Oops. And then also multiple production traits about the tillering, the plant height, all those sorts of stuff as well. And so really that's going to give us unprecedented opportunity to really push yields right to that right-hand side of the that normal distribution of any populations. So that's where we come through with genomics and phenomics, genomic selection and phenomics. I'll change tracks a little bit now and talk a little bit about genetically modified sugarcane. So GM is different from genomic selection. Genetic modification is when you take a bit of DNA from an exotic organism. So say the classic is from an insect that can confer some sort of trait and you put it into a crop plant. That's transgenic. The transgenes come from another organism. And this has been used successfully in sugarcane for Roundup Ready sugarcane. You can see here, this one's got the gene in it. This one doesn't have the gene, and even at quite low levels of glyphosate, we're getting resistant in the one that has the gene. I think I said that the right way around. Yeah. We've done this before, but unfortunately we were in cahoots with a multinational, and they decided they didn't want this work to go forward. So where we're up to now, the reason it didn't go forward is because the international markets were not going to be accepting of Australian GM sugar. So where we're up to now, a little bit of reinventing the wheel, but the international environment's changed. So GM crops currently cover 190 million hectares, or 10 to 12% of global food production. That's pretty impressive. 32 countries actively plant GM crops. Brazil are the first one out the gate. I think Colombia also. They're looking at, Brazil has certainly released a GM sugarcane and have had glyphosate and sugarcane since 2017. There are a range of traits available, stem borer and top borer, along with glyphosate, all in the one genetic construct. And we could look at expanding that to other traits. There'd be a fair bit of research involved in that. And so where we sit with GM at the moment is we want to go back and make sure we do the due diligence this time. Really make sure that the international markets are going to accept it, that we've got access to these traits, free access to these traits, that we can deploy them. And there was also going to be domestic market acceptance too, and that could be for the general community, but also the mills are happy and the growers are happy to grow this sort of stuff. So I'd say within 18 to 24 months, we'll do this this background research to make sure that we have freedom to operate, and if we do, then we can start to look at developing GM crops for Australia. Going to change tracks again now and talk about something that might sound a bit mundane, statistical methods. Now, I'm not going to go into it in great detail, but this is probably the one big thing that I see in our breeding program that can really move the dial. All that genomic selection work I talked to you is based on statistics as well, right? But what SRA has been doing in the past has been doing standard statistical methods, but actually statistics is funnily enough, a very dynamic field, and they're always pushing the boundaries about what are better methods of analysis. I'm working with people out of the University of Wollongong. The guy down there is a global figure in this sort of area, and he provides statistical support for all of the major breeding companies in Australia. I used him when I was in the pulse world and in wheat before that, and certainly the big players in wheat, Intergrain and AGT, also use his services too. So we're going to sort of upskill our staff and our statistical analysis pipeline. And the first thing I'm looking at doing is operating as one SRA. Just have one breeding program. We kind of have 6 regional breeding programs, 4 to 6 regional breeding programs. I'll talk a little bit about that in a minute. That's one way we can improve it. Also doing a combined analysis of 20 fats per year. So it's a multi-environment trial analysis. Currently, we just take, say, the 4 fat trials in the north and we just do a statistical analysis on them. That's okay, but we really should try to combine all of our data sets together. But more than that, we should also go back at least 5 years of this fat trial data. So suddenly the MET goes from our current MET only has 4 trials in one region. Then if we combine all trials together, it'd have 20 trials together. And then if we go back over the 5 years, it'd have 100 trials all in the one analysis. But then we can also go back, and it's really important to do this for various statistical reasons, to go back and get the CAT data from the stage 2 material and bring that into the trial analysis as well. So that'll give us another probably 15 trials as well to put in. So it gets to be a really big data set. But what that allows us to do is really to look at genotype by environment by management interactions, and I'll talk a little bit about that and give you an example of what we've done so far, and it really helps us in variety release decisions. There's also opportunities to improve statistics with the correlation models that we do in combining plant first and second return data. We know that the individual plots are all correlated between those, so we can apply statistical models that can take advantage of that. And another one, and what these graphs are relating to, is looking at how we do our trial design, incorporating a family structure. And what does that mean? It means like if you've got 2 lines that come from the same pedigree, they are related by about 25%, right? And so they kind of behave the same way. It's like you look like your brother and your sister more so than what you'd look like to anybody else in the population. And you can use that in your analysis and your design parameters to improve the accuracy of your trialing structure. And that's what this graph shows here, is that, say, this talks about a heritability of 0.5, which is kind of where sugarcane is. And this is the design method, say at whatever level of replication we've got. That's this value here is when we are doing a normal design like we do, and then this is the advantage we get. Oh God, there's not much there, but actually if that's 0.3 and that's 0.27, that's a pretty big, or 0.28 maybe. That's nearly a 10% jump in accuracy. Right. And what I find with this statistics is I've got all these other little ideas coming down through here, and if each one can give a 5 or a 10% increase in accuracy, they all add up to what I like to call the gold standard. Let's get the best we possibly can. And the reason we should do this is because running yield trials takes up about, I think it's about 65% of my budget, and it's a big budget, 65%. And yet we can do this statistics that's going to increase our accuracy dramatically, and it's not going to cost us hardly anything more, a bit of money for a statistician to come and do the work for us. And so I think I'm really excited to really make sure we employ cutting edge stats on this. And then finally, we've got a research project that's just started out, where we're going to really pull apart the whole trialing system, put costs against it, put genetic gain measures against it, and come up with a bespoke trialing system to really not only fully utilize our genomic capabilities, but also to make sure we can deliver the best possible varieties that we can. So I wanted to talk a little bit about statistics or some results actually, and this is from what we call this interaction class model. It's a new method that's been around for about 3 or 4 years. I've used it in pulses, and we want to put it into sugarcane. And we've just had a bit of a play with it. I did it in the northern trials. So these are northern trials from 2020 to 2024, I think it is. And we've thrown in a couple of trials from the Herbert region as well. And what it is, it's a correlation map showing the ranking that ranks the germplasm of the different trial sites and puts them into some sort of correlation map. So where you've got yellow, it means that the 2 trials that are related here, so it's that one, that's Mulgrave 2320. compared to, what's that one there? Mulgrave 22, 320. They were very highly correlated, right? And then you can put this matrix out there and it's got some fancy stats behind it. But what it says is that all these trials kind of rank the germplasm in the same way. And then you look at what it is. Well, there's 3 trials from Tully over 3 different years and 3 trials from Mulgrave and 1 trial from Babinda. So it's kind of like they're all in the same sort of area. Now, how can we use it in the breeding pipeline, though? We can also pull apart, look at individual clone performance in these different clusters. And so this is a little bit in reverse order. This cluster here is the first one here with 6 entries, and this one I've earmarked here is this second one here. The purple line is the behaviour of SRA 26, a good line in the north, and we've also got Q208 in the red. So there's a couple of good comparators that people might be familiar with. And you can see SRA 26 is dominating here. It's got the best yield potential. But these 2 new clones, SRA 40 and SRA 50, just released this year, they seem to be dominating in this cluster, outperforming SRA 26 and Q208 in this particular cluster. Which cluster is it? It's this cluster of trials here. The interesting thing about these trials is that Tully and Mulgrave are all well-drained alluvial soils, whereas the rest of these trials are not. They're much heavier soils, prone to waterlogging and so forth. So we're saying that, well, potentially this is starting to give us some insight about how this variety performs in this sort of region. So that's great, gives us good information. The other interesting thing about this graph here is that we've got 2 trials from Herbert up here. They were both what we call the Herbert drier trials. They only get about 1.5 litre, 1.5, whatever it is, litres of water rain a year, 1,500 mils of rain a year. So it's drier area, and they've clustered together as opposed to these 2 that have come from the Herbert wet areas as well. So we try to use this cluster analysis to give us some sort of insight. I think we'll leave the questions till the end if we can, Peter, if that's all right. Genotype by environment. So then how does that pan out? What I really want to do is the Met on all trials here that we run right across the site and get a much bigger correlation map of 120 trials over 5 years would be great, you know. But trying to work out what that means in relation to the environments that we grow in. This map here is what we call a Köppen-Geiger climate map. So there's a scientific studies done on different climates around the world, and they put environments into different clusters based on maximum, minimum temperatures, rainfall, when the rainfall occurs through the year, so forth. And then if we put where our trials are, it looks like the North and Herbert all fall in this tropical monsoon region. The Burdekin has this sort of green area, but a little bit of this red that they call tropical dry winter, but we're going to irrigate that anyway, so I don't know what that's going to do to our trials. The central region down here is monsoonal, humid subtropic, and down south in New South Wales, they seem to be in this green area. So it looks like there's probably some environmental factors that are looking at how we do our clustering of our trials, and that's actually been borne out in some research that was done about 15, 20 years ago, where they looked at this correlation analysis and they found that the North coastal trials, the Herbert and the Burdekin, all produced data that ranked the germplasm in the same way. So maybe that's actually just one agroecological environment, and that's being supported by what we're seeing by the climate data. Again, New South Wales and the South as one environment, Central standing out by itself, and the North Tablelands again a bit out there. I'm going to keep on going forward a bit. We're moving through here. So what a new trialling system might look like. Here we've got, maybe we can have a trialling system for the north that includes all 3 areas, for the central, and then for the south. And so that's a best guess at this stage, but we've got to do routine statistical analysis year after year of a 5-year Met to really nail this down and really structure a breeding program around that. I'm just going to skip over the next slide. So I said to you breeding works, so why don't we get these yield gains in the farmer's field? That's that graph I showed you before. We're really not getting any more tonnes of cane per hectare. And what concerns me in a breeding program is we know breeding works, we're climbing this ladder for yield potential, but what if the ladder is up against the wrong wall? So I want to make sure we've got the ladder against the right wall, and really the right wall is how do farmers grow sugarcane? We should be breeding under those environments. So I've been trying to work out what the breeding program might do differently than what a farmer does in the field. I assume everybody's got pretty good nutrition. We do plant our cane differently, and that's just what we have to do. We do stick planting, which puts the cane in a bit shallower, different density. the farmers always do billet planting. So that's something I probably can't deal with. I'm assuming everyone's got pretty good weed control. We try to replicate the soils that most farmers grow under. But the big difference is that we don't go out to very high return crops. We're concerned about the breeding cycle and speeding it up and trying to get it. So we only ever go out to 2 returns, but most farmers grow out to 4, maybe even 5 returns. We also harvest very slowly. We treat our cane with kit gloves, whereas in the field, harvesting contractors come through and they harvest quite fast, and in that process they can actually rip stalls out. So we don't really test our varieties under those sort of conditions. I say we've got to pick up our game to replicate what goes on in the field, but also put a call out to growers to make sure they do the best they can as well. And we know that we don't have a lot of clean seed out there, whereas we always use clean seed, and that can be really detrimental to productivity. And also we always grow into fallow, and these numbers here are a bit of a guess from talking to a few agronomists around the area, but it'd be encouraging to get all farmers to go into fallow as well. And so so I want to grow out, I think the big thing here is go out to a 4 return trialling system. What does that look like? This is what one of our fields might look like at the moment. We have a year of fallow. We'll come in and harvest the plant crop. We'll harvest the first return crop, and we'll harvest the second return crop. And then we'll rotate that all around and plant into here next year and so forth. What we could also do, though, is maybe if we can work with some collaborators and extend that out and get out to 3rd and 4th returns. And I don't want to... I won't necessarily have to harvest this. I would like to get a contractor to harvest or harvest it at speed, as is farmer practice, for the 1st, 2nd and 3rd return, and then come back and carefully harvest 4th return. Now, it doesn't mean we don't get data from these, because we can fly the drones over these, and we know there's a very high correlation between the data we get from drones and the tonnes of cane from hectare we actually harvest. So that's a system that we're starting to put into place this year. What have I done? Also want to improve the agronomic knowledge of new varieties. So we've got an opportunity to work a bit closer with the SRA adoption program. We've got a new general manager of adoption, and we're looking at how we can expand that bit of the work within that portfolio within SRA. I've seen the 2027 district impact plans are explicit in supporting variety management. And so I'll work with these guys to work out how we can incorporate accelerated clones into agronomic trials. Then also I want to meet with industry participants to discuss new clones. And so I want to travel around to all the prod boards and stuff in July and August and try to glean knowledge. I think this is something that's been done by SRA before, but probably dropped, the ball's been dropped a bit since I sort of came on board and trying to pick up the pieces. Looking at the timing and structure of new variety guides, so you might see a bit of a different format for them coming forward. And also looking at the variety development program, how can we increase our propagations of these accelerated clones to make more sticks available to the next level users. So, and part of that might lead into what I'm calling farmer participatory plant breeding. Variety development is really good at doing controlled scientific trials to identify those, incremental gains in yield improvements, 5 to 10% yield improvements. We do that. I showed you the data that it works, and we are getting those improvements. Don't always see them in the farmers' fields. We have to address that. However, I don't really see great structures in place. And again, it's probably just because we've had such changeover in staff in recent times of identifying major flaws. So this would be, we really need to know that if a variety is going to lose out 50% of its yield by 4th return, we really need to know that. So how can we make sure we identify that before we release a variety? And I think farmer collaborations are critical for this sort of stuff. So I'm looking at how we might be able to make some of our accelerated clones available to farmers, approved farmers in 10 to 20 metre strips. Just to highlight, though, that we really do have to be concerned around controlling where the material is and how it gets out. So this material, it's not actually SRA's material, it's the industry's material, but we need to protect it on behalf of the industry. And so we would do this by getting people to sign material transfer agreements. If they want to collaborate with us in this way, we would have to get a document signed. That document would state what you can and can't do with it, limited propagation on your farm, absolutely no distribution of unapproved clones to anybody, and we'd have to put in processes in place to make sure we return data to SRA. I might just leave it there, but that's something I'd be keen to work on. And then we've got a new, I just wanted to have a bit of a shout out to the change, or highlight the change we've had over time. We've had a lot of breeders leave over the last couple of years. I think it's probably about 70 or 80 years worth of experience there. We've got enough experience to see us through in the meantime, but we've got a new team coming on board. We've got a couple of PhD students, our variety officers that we're supporting, and we've appointed 4 new people. And the latest one here is Etta, who's come in from Brazil. So it's a great coup for us. And there's one more, a research scientist, David Hughes. I couldn't get a picture of David Hughes. I did try to do a Google search, but the only images that came up were David Hughes, the comedian, and not David Hughes, the statistician. So, but welcome to David as well. And he'll bring a lot of that statistical knowledge around eye classes. Look, in the interest of time, we have got a concept around fast-track breeding where we might be able to breed a variety in 7 years. I'm not going to go into it at this stage, but I'll just want to finish the talk off. SRA runs a large, complex and scientifically advanced breeding programs. I've got some parameters around here about how many clones we do, what we do, advanced statistics, going through to genomic selection, tissue culture. Some things I haven't talked about, international germplasm exchange, but really just a shout out to all of our staff. We've got about 60 full-time dedicated staff, up to 20 casuals during the peak times in the season. Staff are dispersed across 9 different sites, so it's a real challenge to sort of manage all that, but they work really hard in a really complex and quite challenging environment at times, and just say thanks to them and to let everyone know that we're doing a good job and we're looking to the future about how we can do even better. I'll leave it at that.
Thank you, Garry. That was a very comprehensive presentation. And I think the diversity of the topic that you covered in the breeding actually truly highlight the complexity of sugarcane breeding. And I guess the development of the new tools and methodology and AI actually show a very promising opportunity for the future. So thank you. We'll now open the floor for questions. So if you'd like to ask a question, you can either type it in the Q&A or you can raise your hand and we'll give you, and when you're prompted, we'll actually unmute you, and then you can unmute yourself and then ask your question. And then when you finish, could you please mute yourself again? Thank you. So who has a question? Oh, you were very clear, Garry. No question. Actually, I've got a question.
Good on you, Natalie. That's the way. This isn't the one I gave you beforehand, was it? Just in case.
No, it was like, no. I was just wondering, like, you know, like the way you said, like we actually test for disease in a certain time of the breeding pipeline, and you know, so why are we not doing it from the start? Is it because of the dosage and you know, like I suppose that dosage of like resistance genes? If we do it too early, would you think that maybe later on we don't have enough allele that will actually make the resistant stable?
I reckon that's a great question. It's one of the first questions I asked when I came to this program too. You must be like married to a breeder or something, I would say.
Yeah, maybe.
No, look, in any breeding pipeline, you really want to do those highly heritable traits as soon as you can. And typically disease resistance is pretty highly heritable. We've made changes in the program, or my predecessors have, to try to elevate smut and pachymetra screening early in the breeding pipeline. I would like to go even further and try to bring in some other diseases like maybe orange or brown rust or leaf scald as early as we can. At the moment, those smut and pachymetra, we kind of do at that cat stage, at stage 2. And then all the other diseases we kind of do after we release it. And so it doesn't really help you to make selections to, it helps you to make selections to release or how the variety might perform, but it doesn't help you to, you know, enrich for resistance. So smut and pachymetra, we're doing that. The thing about genomics, though, is that completely turns that thinking on its head. This genomics has the ability to rewrite the whole breeding platform, because now what you can do, if we could have prediction equations for all of these traits, we would not only be selecting for those in our early material that we're genotyping, we'd actually be selecting for the right combinations to give that progeny in the first place, which is something that I expect that we do just through phenotypic assessments of parents, but we can do it in a much more profound way. And you think about all the different traits there. Let's just say, for example, each of those 5 or 6 diseases, if they only had 3 genes, so that's pretty 3 genes for resistance, that's pretty simply inherited. That's 18 or 20 genes or something like that. If you've got 18 or 20 genes segregating in a population, you have to look through 1 million individual plants to find that one plant that's got all 20 genes there. 1 in a million. And that's just your disease resistance. And we haven't even started to look at the more complex traits like TCH and CCS and fiber and all that sort of stuff. So that's just the complexities of breeding, and that's how genomics can maybe help us get over this inability to really test germplasm early on for diseases or highly heritable traits. Great question, though.
There is a question in the Q&A from Daniel Mansfield. How does tissue culture play a role at accelerating deployment of new genetics, and can its role be expanded to assist deploying new genetics? Sorry.
Yeah, no, that's great. Look, I just had a meeting the other day with our team. We were discussing around fast-track breeding pipelines. What I didn't go into just before, unfortunately, I just sort of ran out of time there a bit. And we have looked at that, and my recollection is that if we took some high value crosses, say if you had some genomic design crosses that were the really good ones, the really top of the tree crosses, and what we've done is we've taken the seed from those crosses and we've put that seed into tissue culture. You can surface sterilize the seed, put it into tissue culture and make clones from those seeds and bulk those up so that you can try to get them into yield trials quicker. And you can probably save a year or 2 by doing that, and that'd be great. The other way you can speed up the pipeline, it's not so much the pipeline, but it's getting material out to growers. So if you can identify a clone, say at the end of stage 3, and you know it's got good disease resistance and everything else, we can put that into tissue culture and really ramp that side of the pipeline up. And so to get that product development phase a bit shorter, that accelerate to max crop to release a little bit shorter. So they're the 2 ways where you can fit tissue culture in. Certainly got to fit though.
There's a question from Ben Ashburner. Does the Wilmar program do anything different?
Yeah, the Wilmar program have jumped on. They've got a PhD student there, and they've jumped on the genomics, and they've been using genomics in their parental selection. And so we work hand in glove with the Wilmar program. We swap all of our data. They do some trials for us. They use our fat clones to do trials for us, and we share all of our data with them. We share all the genotyping data with them, and they've been using that to do some parental selections. And a smaller crossing cycle, and by all accounts it's starting to look pretty good. So yeah, so we'll be working with them, and I think they'll be quite happy once we can start to genotype a whole bunch more clones and provide that data to them to help them along. It's certainly a 2-way street, that one.
As there's a question from Azrat. Is the sugarcane resistance to smut is qualitative or quantitative?
That's a good question. Look, I don't really have the answer. I think it's probably fairly simply inherited. No doubt you can always have quantitative resistance if you breed for it. I'm pretty sure we've probably got fairly simply inherited genetics at the moment, and I think that probably leads to another question. Well, could it ever break down? And it could, and we actually are seeing a lot of clones that we think have moderate to high levels of resistance, and yet they're getting quite a bit of smut in the field. That can be overridden by a very heavy epidemic here and stuff, but yeah, I'm not sure. I think it's a good question, and I don't think I'm not over the literature to answer that really detailed, but semi-quantitative, I think.
Anyone else has a question? Because I have another question. No, I would just prefer, like, to give the hands to anyone who has a question, but while we're waiting, because we're going to be finishing 5 minutes, so if you have a question or just make sure you put it in the Q&A or the chat. So my question is, what about the integration breeding program? Like, does the same things that you just presented will apply? Because, you know, I suppose we don't want to lose our diversity. And so the same method can be applied in integration breeding?
Yes. Oh, look, there's the integration program is critical to success of the variety development program. I think we did some work just recently, and 70% of our release varieties or something have at least 70% have at least a grandparent that comes from international germplasm. Maybe that's not integration, but it's international germplasm. But the integration program is critical in maintaining diversity, and we've had success in finding nematode resistance. And so you're obviously very familiar, Natalie, with the work on that. But also we've done some work with integration to get energy canes up and running, and so we've got 15 canes in yield trials at the moment that look like energy canes, and another 15 in the pipeline coming through. So that's great. And if you look at the application of genomics to integration work, I guess there's questions to be answered around how our marker systems will behave in an integration progeny. But it enables you to do targeted selection of the background genetics. So typically in an integration program, you'd make a cross and all the cross looks pretty average. And so then you make a back cross and the back cross progeny looks pretty average, and so on and so on. And you might get out to 3 or 4 back crosses before you start to get decent material. If you can run the SNP chip over the progeny, you can actually select more for the elite parent rather than integration, and hopefully bring your integration material that's useful from, say, some trait-based marker.
There is a question from Megan. Thanks, Megan. You can
How has DPI funding helped the breeding program? And what areas could benefit from DPI funding in the future, I guess? Look, the DPI funding has been instrumental in, look, I haven't been involved in it so much, but really around all of that genomics work. You know, and I'm sure DPI, other people are more qualified, Megan, to talk about that than me. But certainly it's been instrumental in getting that work up and going and showing proof of concept, and there's other research as well. And going forward, and I've talked to Megan about this as well, is that SRA in doing its current strategic plan, you know, really feels like it has to focus on the applied end of the research spectrum. So really getting our technologies out there and investing in making a difference in the farmers' fields. And DPI kind of likes to go the other way, and this is, what's the blue sky stuff? What's going to be the dial changes in 5 or 10 years time, and can we invest in them? So I think it's a really good fit that we can have in that way if we focus our resources on you know, helping the farmers today, and if DPI are happy to keep on helping the farmers of tomorrow, that's a good fit.
Absolutely. I mean, I've got a DPI fully funded project on nematode, and yes, I think it's, you know, it's in the blue sky era. We're doing like great works, and I'm sure that we can all agree that it is very important for us and for SRA. So thank you. I think you know, this is the end. Thank you, Garry, for presenting today. It was a long due. I think we can all agree that there was a great presentation. We had a really good question, and actually the future of SRA is very exciting in breeding. Thank you, everyone, for attending today. I hope you've enjoyed the session. And if you want to find about the new next session, like, you can visit the event page of SRA. We look forward to welcoming you at the new or the next SRA webinar, and we'll announce soon what it's going to be about. Thank you, everyone, again, and have a great afternoon.
Thanks, everyone.
Thank you.
Great.